Hello AP Biology class. Today we will be learning about the cell cycle, but specifically about interphase. So, what is interphase? Interphase is the phase of the cell cycle in which the cell spends most of its lifetime, and also the cell obtains nutrients, grows, and copies its DNA. Interphase is also the longest process in the cell cycle. It is composed of 3 stages. It is composed of G1, or also called gap 1, the S phase or synthesis stage, and finally G2 or gap 2. We'll start out by explaining what goes or what happens in G1 of interphase. So, in G1 we first start with the original cell. The original cell, as you can see, has the same number of organelles and it's only for one cell. But in G1 cell it starts to duplicate. So as you can see, in the G1 cell all organelles are duplicated. This stage is where the cell grows physically, copies organelles, and molecular binding blocks are made. Within the cell cycle, there are 3 checkpoints that must be passed in order to complete successful cell division. Within interphase, there is only 2 checkpoints, and the first we're going to study is G1, which checks for size, nutrients, molecular signals, DNA integrity, etc. The G1 checkpoint determines whether the cell can move on to synthesis phase and if there is need to cell division to be completed. The G1 checkpoint checks for size, where the cell is physically large enough to undergo division, nutrition, where there are sufficient energy and nutrients available, molecular signals, meaning if cells in the vicinity are signaling for cell division, and DNA integrity, whether the cell's DNA is damaged. The G1 checkpoint also determines whether the cell will go on to divide or will go into G0 or its resting stage. Next goes the S phase or the synthesis phase. The S phase goes right after the G1 phase and can only begin if it is past the G1 checkpoint. The most important part of the S phase is DNA replication. The DNA replication is set to produce double amounts of DNA, which means that after this replication, each chromosome consists of 2 sister chromatids. At this instance, the chromosome count in the cell is still 2n. Their nuclei are still diploid, while the number of centromeres and chromosomes remain unaffected from the beginning to the end. The S phase has to happen fast because the unpaired strands are very vulnerable to mutagens that can in the future create complications such as abnormalities. This phase is regulated by various controls, like for example the P16 protein that ensures if the cell is ready to go ahead to the next phase. Next goes the G2 phase, which is the final stage of interphase before mitosis and meiosis. This phase actually happens after DNA replication in the synthesis phase. G2 also has the G2 checkpoint, which actually checks for any errors in DNA and focuses on either repairing those errors or sending the cell into apoptosis, if irreparable, cell death. The base of G2 phase is after DNA replication in the S phase. There is more cell growth, so the cell can have some appropriate conditions for it to start mitosis or meiosis. G2 phase starts off by the nuclear envelope enclosing the nucleus, which contains 1 or more nucleoli. They also have centrosomes, which are regions in the animal cells that organize the microtubules of the spindle. Chromosomes duplicate during the synthesis phase and cannot be seen individually because they have not yet condensed, in which this form they are called chromatins. After the cell undergoes the G2 or the gap 2 phase, then it is all now ready to undergo mitosis or meiosis. Now the cell is ready to undergo cell division.